Good morning. How's everybody doing? Good. Good. I'm David Roebuck. I'm the Senior Vice President for Academic Affairs. And I apparently broke a microphone. Senior Vice President for Academic Affairs and Dean of Faculty. And I'm honored to welcome you here this morning to the Undergraduate Scholars Forum. This is the 15th Westminster Undergraduate Scholars Forum. And I got to tell you, I've been in higher education for quite a while. This is one of the coolest, most unique things I've ever seen on a college campus anywhere. And I'm really proud to be part of it here today. Today Westminster students have the opportunity to demonstrate the things that they've learned in the classroom and apply those to real circumstances. Today we highlight the creativity and scholarship, other activities of Westminster students. Today our students explore topics from math and science, business and social sciences, humanities, from almost every academic discipline on campus. Today we give our students yet one additional unique opportunity to flourish. Today is truly a great day. Today is truly a great day. I'm glad to have you here. I'm glad to have you participating in Undergraduate Scholars Forum. My sincere thanks to the students for all the hard work that you put forth to make this possible, and my sincere thanks to the faculty for serving as outstanding mentors for these students. Again, thank you. Have a great day. And with that, I will turn this over to Dr. Carolyn Perry, who will recognize Dr. David Stubbs.
Good morning. Good morning. Back in the fall of 2007, I was the division chair for the humanities, and we were sitting around a table with the then Dean of Faculty, Barney Forsyth, and he was telling us about this great idea he had. He wanted to launch in the spring of 2008, called the Undergraduate Scholars Forum. We were all like, go Dean Forsyth, that's a great idea, you do that. Little did I know that a few weeks later he would become the president of the college, and I would become the Dean of Faculty, and the Scholars Forum would be added to my plate instead. And so I had the great privilege of getting to launch the first Undergraduate Scholars Forum back in 2008, and I have treasured this day ever since. We'll try again. One more. I don't mind projecting. Okay, all right. Shortly after we launched this event, Dr. David Stubbs noticed that this was an important thing on our campus. And what he said was, it's great to have this, but we need to make sure students know how much we appreciate their great work. And he asked that we start adding cash prizes to those students who come forward with the best presentation, best posters, to show off their great work. And the Stubbs family then rallied around this event and actually pulled together an endowed account for the Undergraduate Scholars Forum, so that this event can be part of our campus, part of our academic culture, virtually forever. And that's an exciting thing. We're very thankful that the Stubbs family saw that this was an important event and did everything they could to make sure that it will be here for you from now on. So I wanted to take this time to recognize the Stubbs family, but Dr. Stubbs in particular, because of their vision for this event. And on the 15th anniversary, it seemed like a great time to recognize the family. And so I would like for him to come forward and just say a few words. Dr. Stubbs graduated from Westminster in 1968 with a degree in biology, and he went on to med school at the University of Missouri, specializing in vascular surgery. He has practiced medicine in Iowa since the early 1980s, but I wanted you to know that he's not just a renowned surgeon. He's also a professor of surgery. So he understands the value of teaching and the value of what we're doing here, and I find that just wonderful. Westminster is very proud of all that Dr. Stubbs has done, and we recognize his success in 2008 with an Alumni Achievement Award. We're very thankful, Dr. Stubbs, for your continued support of the college, but in particular for your support of this event. So please join me now in welcoming Dr. David Stubbs.
President Lofi, Dr. Perry, Dr. Madras, and in honor of Dr. Fickus, Dr. Williams, Dr. Schulte, who were my professors back in the day, we welcome you to the Scholars Forum. We certainly appreciate the opportunity to support this unique event and hope that it is a unique opportunity for the faculty to support and encourage their students to pursue creative projects and research that may well light a spark that will become a life passion for each of you. We hope that your participation today has a unique sense of accomplishment and will be encouragement to continue pursuit of your dreams. My family and I thank you.
And now I have the great pleasure of introducing you to another Dr. Stubbs, and this time our own Rebecca, graduate of the 2008 Westminster class. Dr. Rebecca Stubbs is currently in a postdoc position with the Department of Systematic and Evolutionary Botany at the University of Zurich. Rebecca's current work at the university focuses on genomics to study evolutionary biology. Prior to her postdoc position, Rebecca earned her PhD in biology in 2018 from the University of Florida, Gainesville, her master's degree in biology in 2012 from San Francisco State University, and her bachelor's degree in environmental science with a minor in chemistry from Westminster. Rebecca has published extensively, she has made numerous presentations, and she's received several grants to support her work. Among her awards is the 2018 George R. Cooley Award, which is among the most prestigious awards given in the plant sciences. Her passion for the study of plants led to the discovery of a new plant species in Northern California. Think about how cool it is to have your name, the Stubbs name, on a new species of plants. That's pretty cool. Dr. Stubbs is a lifelong learner, and she is one of our own. We are so proud and pleased to have Rebecca with us from Switzerland today. Please join me in welcoming Dr. Rebecca Stubbs.
All right, I'm presuming you all can hear me, otherwise someone will let me know. Thank you for the nice introduction and to everyone here for attending my talk. I am honored to have been asked to speak at the Undergraduate Scholars Forum, and I'm glad I was able to virtually attend from Switzerland. I am very excited to be presenting both my research and my career trajectory to all of you at Westminster today. I realize there are people in the audience who do not study science, so I'm going to explain what I do and the background needed to understand my research using relevant examples. I will then segue into my research and talk about my journey from being an undergraduate at Westminster to a postdoctoral researcher in Switzerland. I study evolutionary biology, which you may not think you know, but in fact you have heard about. The classic example is Darwin's finches, which have a remarkable diversity in beak form and function as a response to the different foods that the finches ate. But why is evolutionary biology important? It is the study of the evolutionary processes that produce the diversity of life on Earth. Evolutionary biology is the product of being curious about the natural world and all of its organisms. And to explain how one conducts evolutionary biology research, I will start with something we can all relate to, which is a family tree. So here we have the family tree of Winston Churchill. You can all picture your own family trees in your head with your siblings, cousins, parents, aunts, and uncles, and grandparents. In this case, you are picturing individuals. I could ask you about who are your parents, grandparents, etc., and we could make a family tree like Winston Churchill's. But the first step away from family trees towards thinking evolutionarily is to think of family trees not just with relationships, but instead where your genetics come from. So you would be generation 0, and you could work back in time identifying biological parents. So now we are thinking biologically, but in the case of family trees, we are still thinking about individuals. One of the amazing things about evolutionary biology is that not only individuals, but species, genera, families, and orders can also share family trees. I found these pretty family trees of different bee and seahorse species, but really you can make a family tree for any organism. This interest in how all living things are related goes back a long time. And in fact, I just mentioned Darwin. Many of you have heard of his book, The Origin of Species, which is considered the beginning of evolutionary studies. Well, there is exactly one and only one drawing in that book. It is Darwin's attempt to understand how species are related and evolve. And it very much looks like a family tree, which shows that people have been thinking about and curious about evolutionary biology for centuries. We can figure out family trees through records or simply talking to people, but how do we know who nudibranchs or orchids or the Swiss ibex or COVID is related to? We can't just ask them. The answer is DNA, and we've all seen these pictures of double-stranded DNA and single-stranded RNA at some point. But for our purpose today, what is DNA? I realize most people have heard about DNA in biology class, but I just wanted to give a short recap. It is found in chromosomes inside the cells, and it is in every living thing. DNA determines such things from eye color to how organs function. Each piece of information is carried on a different section of the DNA, which are called genes. The four chemicals that I'll refer to by their initials of A, T, C, and G that make up DNA are repeated in different orders over and over again in each strand of DNA. Human DNA contains about 3 billion pairs of these bases. The order in which the bases are arranged is very important. It forms a code that tells cells to make certain kinds of proteins. The differences in these proteins are what makes different living things, such as cacti, gerbils, and even two different people, different. Finally, a genome is all of the DNA in an organism, and to use genomes in research, we need to know the order of those A, T, C's, and G's. Why does this order matter? Well, one example is that humans share well over 90% of their DNA with their primate relatives. Even though we are remarkably different species, we have remarkably similar genomes. In fact, if you look left and right, the genetic difference between you and the person sitting next to you is only 0.1%. So what do genomes have to do with family trees? Well, we can use DNA to understand how species, both living and extinct, are related through identifying genetic changes. And this is the field of evolutionary biology. We use a sort of family tree with DNA to understand evolution. So far, we have talked about family trees, which includes individuals. I then showed you Darwin's tree, which was to understand how species are related. Our new term is phylogeny, which for our purposes today is simply a tree generated from DNA. It tells us similar information, but instead of being created from observation, it uses genetic data. You will also hear me say phylogenetics, which is simply the study of phylogenies. And tree and phylogeny are in fact interchangeable terms. I will be using both through the rest of this talk. You may have never seen a phylogeny before this talk, so I'll briefly explain how to understand it. Everything at the edge of the tree represents a species. They're each sitting on a branch. In this case, the branches represent time, but they can also represent differences in the genome. There are also branching points that split into two. Any two species that share a branching point are more closely related than any other species and are called sister species. So Homo sapiens are sister to Neanderthals. And at this branching point is the most recent common ancestor of humans and Neanderthals. Humans did not evolve from Neanderthals, but we share a common ancestor with them. And in fact, this is part of a much larger phylogeny of all human species and our ancestors. And here we can see that we share an ancestor with apes. We did not evolve from apes, but we do have a common ancestor. We've been introduced to DNA and now phylogenies, but how do you go from DNA to a tree showing how we are all related? The keyword in this sentence is, the way to think about this is that as DNA passes through the generations, it changes through mutation so that it varies among individuals. And the keyword in this sentence is mutation. These mutations are key to understanding which organisms are more similar. The most similar organisms share all of the same mutations and have the overall most similar genomes. One way to find mutations is to use a PCR machine. If you had told me 10 years ago when I was first learning how to use a PCR machine that the world's most famous soccer player would have headlines in 2020 discussing his thoughts, albeit incorrect thoughts, on PCR, I would never have believed you. But now everyone has heard of PCR. So I've updated Ronaldo's quote to be more accurate because, in fact, PCR machines are remarkable scientific instruments. And even before COVID, PCRs were considered one of the most important inventions for research. In fact, the inventors were given the Nobel Prize in Chemistry in '93. Briefly, how a PCR machine works is that it looks for a bit of DNA or RNA, and if it finds what it's looking for, it makes a ton of copies of it. From there, you can either just look for presence or absence, as in the case of COVID test, or sequence it, which means get the genetic code of the DNA. Sequence data, you can look for mutations. Now, this figure is supposed to represent 201 positions of the COVID genome, but I didn't feel like making that many rectangles, so just pretend with me that it's 201. And here's the original strain of COVID. In these 201 sites, there were 2 mutations in the Delta variant, so not that different from the original strain. But then Omicron comes along and we have 15 mutations. This was enough mutations that in some cases, this variant was able to get past any previous immunity. Scientists have documented that of these 201 sites, there are 2,000 different ways to mutate, and 400 of which have already been seen. So you can use all of these mutations to understand how COVID genomes are related. So why is this important? Here is a phylogeny of 3,000 COVID genomes representing all known variants starting from 2020 and proceeding until last month. You can see on the left side of the screen the date range. So this phylogeny is showing the evolution of COVID for the last 2 years. This is from this website called NextStrain, and I put the web address in the corner if you want to check it out for yourself. This is the same as the other phylogenies we have looked at, but it has 3,000 branches, so it is a bit harder to see the branching pattern. Every little dot is one genome, and using computer programs, we can look at mutations in each individual virus and make a phylogeny from this. Branch lengths represent variation, so if only a few base pairs are different, you get a short branch. But when Omicron comes along, which are the orange and red groups at the top of the screen, you will see they are sitting on very long branches because of all of the mutations. And of course, we are all familiar with COVID, which is why I started with this example, but phylogenetics, in fact, has many applications that are directly tied to our everyday lives. So now I will briefly give one more example of genomics and evolutionary biology, but using something closer to my heart, which is, of course, plants. You may not be aware that all of the food you eat has been heavily selected for over centuries and has very narrow genetics. And I'll explain in a second about narrow genetics. But farmers around the world have recently experienced significant crop losses due to severe heat and drought. Such extreme weather events and the need to feed a rapidly growing population has raised concerns for global food security. While plant breeding has been very successful and has delivered today's highly productive crop varieties, the rate of genetic improvement must double to feed the world in the near future. So an extreme example of the problems with crops with narrow genetics is the Irish potato famine from the 1850s. What caused the famine is that the potatoes that sustained the population of Ireland stopped growing due to a fungus. Why did this happen? If you've ever planted potatoes, you know that you don't need to grow a potato plant from seed every time. You can break a potato in half and put it in the ground. From this potato chunk, a new plant with more potatoes will grow, which is how they propagated potatoes in Ireland. When you do this, though, you are simply making identical clones. These are all genetically identical individuals. The problem was that all of these clones were all susceptible to potato blight, thus resulting in the famine. There are potatoes that naturally carry the genes that are resistant to the blight. And if there had been more genetic diversity, it is widely believed that the impact of the blight would have been less severe. And cloning potatoes is the extreme version, but in fact, all crops are considered to have narrow genetics. The reason for this is that in the beginning, for each species, there were a lot of differences in their DNA and a diversity of genetics. But humans selected for very specific traits such as taste or size over centuries and then propagated those same plants until we were left with monocultures of almost identical plants. Consequently, all crops have hugely reduced genetic variation. And genetic variation is essential for new traits in dealing with climate change. One solution to low genetic diversity is to find the closest relatives to present-day crop species. Across the top of this figure is the eggplant crop that you buy at the store. Humans selected these for color and size and taste. Now, for every crop, there is an undomesticated wild relative somewhere out there in nature. And you may not have known that, but if you think of any fruit or vegetable from apples to zucchinis, there is a wild undomesticated version of that crop that will look a little different and likely not taste as great, but it is out there. One of the things you can do when you identify the closest relatives to a crop is that they can be crossed to make hybrids. These hybrids will have the genes that we originally selected for, such as taste and color, but will also have new genetic variation mixed in from this wild relative. So how do you know which species to cross to make hybrids? Well, the best case scenario is to use the closest sister species. To do this, you need a phylogeny with your crop of interest and all of the wild relatives. This example is for pumpkins and squashes, and you can see that there are a bunch of groups that have both wild species and crops, and these would make good pairs for crossing to increase genetic variation. Okay, so that ends our science class for today. Hopefully you all now have a basic idea of what is evolutionary biology. I'll now move on to talk about how I got to where I am today, including sharing a few of my adventures from fieldwork to give you a reprieve from all of this DNA and phylogeny talk. And this journey begins right here at Westminster. You may have read that I am from Iowa, so my first big move was a 5-hour drive south to Fulton, Missouri. Now, these are the obligatory Westminster graduation photos, and you will all have these too. You with your family in front of the columns and a picture of you walking through the columns. I'm sure I'm in there somewhere. I graduated with an environmental science major. One of the things about my degree from Westminster is that when I was at Westminster, I needed an internship to graduate. I got this cool internship working for the Iowa Department of Natural Resources. This internship was paid and had me outside quite a bit, which at the time was not something I thought I would like, but I ended up loving it. I had almost no experience, but the reason I was able to get this internship was because I had participated in service learning courses at Westminster, and I used those experiences to bolster my application. Another great thing about my time at Westminster is that I participated in a travel course to Costa Rica with the Environmental Science Department. Additionally, I spent a semester studying abroad in Spain, thinking I could improve my Spanish, of course not knowing that one day I'd be living in a German-speaking country. In hindsight, these sort of experiences end up being fundamental to where I am now. It's important during your undergrad to be involved and take advantage of these opportunities. And I will circle back to some pivotal moments from my time at Westminster at the end of this talk. But first, there's one very important story involving Dr. Amsboker. I know that Dr. Amsboker retired in 2019, but some of you may still know him. In 2008, I was close to graduating and was still unsure what to do. So I went to talk to him, and I knew I was open to grad school and that I liked science and I liked biology, but not really humans. And by that, I mean, I didn't want to go pre-med. I still get queasy at the sight of blood. And so he listened to my story and said, well, what about plants? And in fact, I'm offering a botany summer course. Why don't you try it? And I did, and it set me on the path that I'm on now because I subsequently applied to grad school in botany. I'm going to do these little recaps at every stage to comment about how I thought of myself occupationally and also what my interests were. So I graduated and I was an environmental science major with a minor in chemistry. I knew that I liked the outdoors and I liked science, but other than that, I was still figuring it out. So I applied to graduate school and I moved to San Francisco to attend San Francisco State University. And here I am looking embarrassingly like a hipster with my master's advisor, but my first year, I was basically a postbac and I took quite a few botany, biology, and ecology courses. Well, over this year, in addition to my classwork, I was supposed to be deciding on a project to study for my master's. And that year came and went and I didn't have anything. So a friend hooked me up with a job near Yosemite building trails. It's a beautiful area of the world. It was a cool experience, and I now know how to use a chainsaw, but it was not exactly a science job. But there was an important event that happened that summer, which was that I was invited on a hike to climb a mountain. In fact, it was my first mountain. I think it took 15 hours and there wasn't a trail and we got lost at some point, but I loved it. It really opened up a whole new world for me and started a now lifelong interest in summiting mountains. But most importantly, up near the top, I saw this small plant with bluish purple flowers and funny little leaves growing by itself higher up than any other plant. I snapped a picture of it with my first digital camera and didn't think about it again until I climbed my second mountain. And there was this plant again with bluish purple flowers and funny little leaves and growing higher than any other plant on the mountain. So I went back to my master's advisor and asked, what about doing a project with this plant, which is called Sky Pilot, and the Latin name is Polemonium. And luckily, my master's advisor was very supportive of this idea. So I had a fun summer of fieldwork and climbing mountains and collecting plants. Then in the fall, I would bring the plants back to the lab. I would then extract the DNA and use the PCR machine to get the DNA. In 2010, when I was doing this, I was just trying to get one gene, and getting a good sequence for one gene for an organism where you don't know the genome took over a year. So I finally got my data and I built my one gene phylogeny, and I ended up with a few cool results. One was for a sky pilot that grows on Mount Shasta in California. This Polemonium looked different from closely related species, and the phylogeny suggested it could be a separate variety. Additionally, this plant grows in an area of the mountain highly impacted by humans. So I described this as a new variety, and I named it after the mountain. So it's variety shastense. Additionally, up in Northern California, there was another sky pilot that was identifiable as different from the other species and grew 100 miles from the nearest population. This species was also growing on a mountain called Mount Eddy that there was some interest in mining. So I described this as a new species and worked with CNPS to get this plant protected as rare and endangered. And in fact, this population is the only known population of this species in the world. And again, I named it after the mountain it was growing, which is Polemonium eddiense. So for my master's, I got to do fieldwork in California and climb mountains. As I was finishing my degree, I found I had a lot of questions about mountain plants. And a lot of those questions had to do with my background in environmental science. The habitats these plants grow are at best unfriendly. So the big question is, why grow here? 1,000 feet below where this photo was taken is a lush valley with tons of plants. Why grow where most of the year the temperature is below freezing, pollinators are scarce, and wind gusts are strong enough to knock me over? I decided to get a PhD with the help of my advisors at University of Florida. I designed a PhD project to address 3 questions. What caused the distribution of alpine and Arctic plants? What's going to happen to cold adapted plants in the future? And what is driving speciation? So another little recap for my time at San Francisco State. At this point, I identified as a botanist/biologist. I was interested in and had a knack for fieldwork and hiking for days to collect plants. And I had a basic understanding of genes and genetics, but I really wanted to prove upon my skills in genetics. So I applied for a PhD program that would allow me to be at the forefront of genomic work. So I moved from California to Florida to attend the University of Florida. And while there, I was studying rock rankers. The Latin name is Micranthes, and most people may not be familiar with this plant, but a few species do in fact grow in Missouri. I chose to work with this group of plant for 2 particular reasons. 1, not much was known about the approximately 80 species in the genus. And 2, in the lower right-hand corner of the screen, I have 2 pie charts. 1 is for all plants on Earth, and of all plants, only a measly 4% of plants are able to grow in either the high mountains, referred to as alpine areas, or high latitudes where the Arctic regions are. But for Micranthes, on the other hand, a whopping 3/4 of the species occur in alpine and Arctic regions. I spent 3 field seasons trying to collect as many Micranthes as I could and covering a large swath of the northern hemisphere. And I know people may not be as familiar with this plant, so here are some of my better pictures of rock breakers. These photos are a bit misleading because if you recall, the Latin name starts with micra, which is due to these plants actually being pretty small, but they are pretty nonetheless. So now you know what I was looking for, which are generally pretty but rather small plants. And my first year, I crisscrossed the lower 48. I went to the Appalachians in North Carolina, the Rockies in Montana and Idaho, Sierra Nevada and Cascade Ranges in California, Oregon, and Washington. I also made quick little trips to Arizona, Arkansas, Missouri, and Nevada. I spent most of the summer camping, and in the upper left-hand corner, you can see my makeshift campground office for processing my collections. The next year, I went to China for the summer, and I had lots of amazing collaborators, a few of which are pictured here. In addition to fieldwork, I had some very cool experiences. For example, the bottom middle photo is from a day off collecting when I went to the Panda Research Facility in Sichuan. Also in the lower right-hand corner, I went to a national park up in the northeast side of China. So on the far side of that lake is North Korea. So I waved at a safe distance to North Korea. I was there for 2 months, and it's hard to choose just one story, but a highlight has to do with the top middle photo. I was collecting micranthes in an area of China that has Tibetan macaques, which are the largest monkeys in Asia. There are all these signs warning tourists about the monkeys and that they're aggressive and you shouldn't feed or mess with the monkeys. Well, I was holding a bag that had a rock breaker in it that I had just collected, and a monkey ran up, grabbed the bag from my hands, thinking it was food, turned around and hissed at me and showed me its fangs. I had traveled across the world to collect this plant, and the species was actually super rare, and this park was the only place in the world it grew. But a quick cost-benefit analysis convinced me that my plants weren't worth getting bit by a monkey for. So I was beside myself at this point, but luckily we had the park botanist with us, Mr. Lee, who was in the photo, was able to distract the monkey with his walking stick and grab my plants back, which he then carried the rest of the trip. So that is the time a monkey stole my plants. The next year, I went to Alaska for the summer, which was a dream come true. The top middle photo is everything I had to carry in my backpack to go into the backcountry of Alaska. My PhD advisor had bought an emergency locator satellite beacon for me, but even with that, who knows how long a rescue would take. So it was always good to be prepared. Luckily, I never needed it. And also, I know when most people go to Alaska, they show pictures of moose and bears, but in the right bottom corner, I have a picture of a muskox, which is like a smaller, fluffy buffalo, and was my favorite animal in Alaska. One trip I did that was particularly cool was when I was flown into the backcountry with the National Park Service and left to hike out and collect plants on the way. We were flown in one at a time, and we had to wear the National Park Service issued flight suits. We spent 3 days and 2 nights backpacking back to town, and this was probably one of the coolest experiences of my life. Talk about true wilderness. There were no trails, no cell phone service, no people, just us and a map and a few cans of bear spray. It was also an extremely successful collecting trip because in all of Alaska, there are 17 micranthes, and on this trip, I collected 10 species. I know that the fieldwork looks fun, and it was, but that was only during the summers. The vast majority of grad school was not doing fieldwork, but I feel very fortunate to have had those experiences, and they're always fun to share. It is important for me to point out that the other 9 months, and then the 2 years where I didn't do any fieldwork because I had all of my plants, were spent taking courses, doing lab work, writing grant proposals, applying for collecting permits, which can be a real ordeal, and analyzing data. Also during grad school, I was teaching, mentoring undergrads, and volunteering with outreach and service projects. And fun fact, during my PhD, my office didn't have any windows. So for all of the hours I spent in my office cave, at least I could use my fieldwork photos to make nice screensavers. And what did I do with all of these collections? I needed the DNA. This involved drying leaves and extracting the DNA, getting DNA ready for sequencing, and then shipping to a sequencing company. When they are done, they send me back terabytes of data. And from this data, I can then run analyses. So of course, one of the first things I did was make a phylogeny. And this phylogeny was made with over 500 genes. You remember my master's phylogeny was only 1 gene. And what I want you to know about making a phylogeny with 500 genes is that it takes a lot of computational power, and storage. A personal computer can't deal with this quantity of data. For instance, the computer I'm giving this presentation on has 500 gigabytes storage, and to simply open the genetic data, I would have needed 4 of these computers. So it is all stored on supercomputers, and to interact with supercomputers or HPCs, high performance computers, you have to use scripting and coding. which is the field of bioinformatics. And with my PhD is where my interest in bioinformatics and computer languages really started. So some general findings from my research is I found new populations for rare plants just because I did so much hiking. I also provided the first genetics for quite a few species. And I did find one new species, hasn't been named yet, but I'm working with the national forest that it grows in to make sure it is protected. So that brings me to my first question. What caused the distribution? One of the cool things you can do with phylogenies is run models on them to make predictions about what was happening in your group of interests in history. For instance, Micranthes diverged 60 million years ago, which is shortly after the KPG extinction event when the dinosaurs went extinct. Another big date for Micranthes was when the plants moved south to the contiguous United States around 37 million years ago. This was during the Oligocene, which was a relatively cold area where cool adapted organisms are believed to have spread around the world. Instead of showing this on a phylogeny, I can also show this on a timeline. First, I can add in the important dates for Micranthes, and then add in the mountain building events and major climatic changes. And there seems to be a pattern of major disruptions of ecosystems and then bursts of diversification in Micranthes. So it seems while other plants and animals were going extinct, Micranthes was thriving and taking advantage of these times of low competition. And this is a pattern we still see with rock breakers growing where no other plants like to grow, where competition is low, for instance, out of the side of a mountain, hundreds of feet above the nearest water source. So for the next question, what will happen in the future? To answer this question, I need to know about a species niche. Niche describes all of the variables in an organism's immediate environment. And remember, I have a background in environmental science from Westminster, so I had a vested interest in investigating the impacts of climate change. Briefly, you can use a plant's niche to understand, plant's niche to investigate climate change through using machine learning. And these analyses are called ecological niche modeling. How this works is that a computer takes a point and collates all of the temperature, precipitation, geology, and elevation data. It then does this for every single point for that organism and uses this to create a model of what kind of condition this plant likes. So let's look at this in real data. This is for Micranthes apetala, which only grows in the high elevation areas of California. All of the yellow dots are all of the recorded collections of this plant. I run some scripts and I get all of the environmental data for each point. Then this data is projected onto maps that have present day conditions. So although this plant only grows in California, the model suggests that there is suitable habitat for it up in Oregon, which makes sense. And if I wanted to find a new population, this would be a good place to start looking. So next, this data is projected onto future conditions representing global warming. This plant loses almost all of its habitat and is predicted to go extinct under the most severe climate change conditions. And that was for one plant, but in fact, I ran these models for months for about 30 species of Micranthes and ended up with tens of thousands of data points. And to summarize the results, with future climate change, 72% of Micranthes would lose their habitat, and the loss of habitat was most pronounced in Arctic species and also high elevation montane species. So to summarize my time at University of Florida, at this point, I considered myself an evolutionary biologist, and I was particularly interested in bioinformatics, scripting, and coding, and I was pretty well versed in genetics, but I knew I wanted a postdoc where I could really improve my bioinformatics skills and have access to large genomic data sets. Which also brings me to my final question, which is what is driving speciation? So this work is being done with primroses, which you may recognize as a garden plant, but they are native to Europe. And this work is, of course, being done in Switzerland at University of Zurich. You can't talk about Switzerland without showing pictures of the Alps. And just remember here that I am from Iowa, a state that's highest elevation is barely over 1,000 feet. And now my hobbies include mountain biking and mountaineering and trail running and all things Switzerland. And if you had told my 18-year-old self that this would be what I'm doing now, I would likely have not believed you. Being here has been a really great experience. And I also feel lucky that I have been working on a very cool project. So back to primula. There is generally this idea of species as being reproductively isolated. And the term for that is species barriers. And we can imagine species barriers as little impermeable walls around each species. But in primroses, there are leaky barriers. And in fact, these species hybridize easily in nature. And this happens in all 3 species pairs. So to understand what is driving speciation, I'm investigating the genomic component of what genes transcend species barriers. To do this, I did field work in 7 countries and collected around 900 samples throughout spring and summer of 2019. And again, I had amazing collaborators and a lot of help along the way, and these people were invaluable to this work. I went to a lot of places for this field work, so I thought I would just show some pictures from the countries of Georgia across the top and Turkey on the bottom, 2 countries I may not have had the opportunity to visit if it wasn't for field work, and they were both spectacular with wonderful culture and food and nature and especially people. In fact, the lower middle picture is from the day that in exchange for helping some local Turkish farmers herd their cows to pasture, they led me to a population of a rare plant, which was quite the day. So for my master's, I worked with 1 gene. For my PhD, I used around 500. And now for this project, I'm working with entire genomes, which is both a dream because I love data and a nightmare because it is a lot to deal with. And of course, I made another phylogeny, but this one is used to understand which species have the most similar genomes. I then use this information to look for specific genes and to address big picture questions about how genes move around between species. So we are working on publishing this data, so I can't say much more about it at this time, but stay tuned. So to bring my presentation to an end, here is a summary of my trajectory from 2008 when I graduated from Westminster to now. Even though now my interests are in bioinformatics and genomics, this all started with a degree in environmental science at Westminster. And in many cases, I kept returning to what I had learned during those 4 years. Now I know there are students in the audience who know what they want to do and they have a plan, and that is great. But for those of you who don't, do not worry. In 2008, bioinformatics was not even a degree, and now it is a highly sought after skill set and my favorite part of my job. Also, the first ever draft of the human genome was completed in 2003, and that took over 10 years and cost $1 billion. So the idea that less than 20 years later, I would have a whole project based on the complete and high quality genome of an obscure plant that most people haven't heard of was unfathomable. So focus on what interests you and understand there are possibilities that you haven't thought of. Finally, take advantage of being at Westminster and all the unique opportunities you have here. I have a degree in environmental science, which was fundamental for my career development and sparked my interest in scientific research. I'm so excited about the Undergraduate Scholars Forum because it allows students the opportunity to explore and celebrate scholarship and research, which is something I'm very passionate about. Also think back to that conversation with Dr. Amsbooker I told you about. In hindsight, that was a pivotal moment in my life. A great thing about Westminster is the small class size, so I would implore all of the students here to engage with the faculty. I also want to give a shout out to Dr. Hansert. I read he is now the chair of the chemistry department, but he had just started a few years before I took his classes back in 2010. I really liked chemistry labs, so I kept taking chemistry classes and I ended up with a minor. Full disclaimer, I do not have much of an aptitude for chemistry, but that was my first real introduction to lab work, which is something I have now spent hours and hours doing. I had no idea at the time how practical getting that minor would end up being. I looked at my transcript when preparing my slides and was surprised to see that I took British literature before 1800. Now, like no shade to pre-19th century Brit Lit, but the classes I am good at are generally science classes. So what was I doing there? And the answer is I was learning how to write. I don't know any field where being a decent writer is not important. From sending a cover letter to a future employer to the dozens of grant proposals and scientific manuscripts I've had to write since leaving Westminster, practicing writing is always important. The other thing I have to do is a lot of presentations. This last summer alone, I spoke virtually at 4 different conferences. Public speaking is hard, but is one of those things that truly gets easier with practice. Well, as is probably predictable for a science major, I definitely struggled a bit in political science and philosophy classes. But I remember having to give a lot of speeches and presentations, and that is the best way to start improving public speaking skills. So I'm now very happy I took those classes. It's important to remember when pursuing scholarship, whether that is psychology or business or education, that it cannot be all consuming. I'd like to point out that although I sometimes work 60 hours a week, it is critical to make time for yourself and do things that bring you joy. I haven't always lived in the mountains, but I've always made time for activities and friends and family, and it is essential to have balance in your life. Westminster has always encouraged students to be well-rounded, so make the most of your time here. I didn't know what to put this last category under, but I was looking at the Westminster website and I saw one of the tenets was global perspectives. When I was at Westminster, there were quite a few international students, and I could have took classes like world religions and Native American spirituality. But truthfully, it took many years to truly appreciate how important this is. During both my graduate and postgraduate experiences, I found myself interacting with people from dozens of countries throughout the semester. In fact, my current lab has 7 people from 7 different countries, and I live in a foreign country, a continent away from home. Being able to interact with people with different backgrounds than yourself and seeing their perspective on the world has not only helped my career immensely, but has enriched my life more than I could have imagined. There are lots of options with a degree from Westminster. Just make sure to be involved and take advantage of new experiences and opportunities. And with that, I would like to thank a few people. First, I'd like to thank Mary Majerus and Carolyn Perry for inviting me to give this talk and helping me prepare. I would also like to thank all of my professors at Westminster, many of whom have retired, but they made a lasting impact on my life. I would also like to thank my family, some of which tuned in from California on the Zoom, and especially my parents who are here today for all of their love and support through 3 degrees and all of my adventures. Also to my siblings, Thad, Courtney, and Zach, for being supportive of me, even when I moved to a different continent. And shout out to my nephew, Jack, who attended my talk today. I especially need to thank my sister, Courtney, who dutifully watched 2 versions of this presentation. And I would like to thank my dad, my brother, Thad, and Beth McClellan for establishing an endowment for the Undergraduate Scholars Forum. Finally, I need to acknowledge all of my graduate advisors and institutes that supported all of my research. And with that, I'll leave you with a nice mountain glacier picture I took from a summit in Switzerland last summer, and I would be happy to take any questions.
Rebecca, we cannot thank you enough for that fascinating and absolutely inspiring talk. Thank you. Thank you. In the interest of time, if you don't mind, I'll have students email you with questions. And we hope you can stay on. We have a few more things during this session I think you would enjoy. And we're just delighted to have you. Again, that was amazing, inspiring, and thank you. Thank you very much. Okay, great. I would now like to introduce our student presenter, Anna Kessner. Anna is a senior from Russellville, Missouri, majoring in mathematics and business administration. Anna is a member of Alpha Chi, Skulls of 7, Kappa Alpha Theta, Blue Blazers, and the Honors Program, among her many activities. She has recently accepted a job with Wells Fargo after graduation. Among our many accomplished students, Anna was selected to give the student presentation this morning. So please join me in welcoming Anna Kessner.
Hello, thank you all for coming. Thank you, Dr. Perry, for the introduction. For anyone who doesn't know me, my name's Anna. I'm a senior this year. If you're wondering if I'm ready for graduation, I'm not, so don't ask. Today I'm going to be talking about psychological safety and how it makes teams more effective. My goal for today is for everyone to walk away as a better leader or a better team member. I'm not a professional, so if you walk out feeling the exact same, you can't blame me, okay? So first off, you're probably wondering why psychological safety, or what even is it? So by a show of hands, how many of you have heard the term psychological safety before? Okay, so not a lot of us. 6 months ago, I hadn't heard of it either. I'm in the honors program, so we're asked to do a senior thesis project, and really the only guideline we're given is that it needs to be interdisciplinary. So I'm majoring in math and business, and then I'm getting a minor in leadership, and I love all 3 of them equally. So I wanted something that I could kind of tie all 3 into. Oh, okay, I thought I did something. Okay, so whenever I was searching for my topic, I tried to think of something I was passionate about. Whenever I started doing research on what makes teams effective, hoping that the answer would be female leaders, it wasn't. I kept stumbling on psychological safety. It kept popping up in a lot of different research that I did, so that's kind of what I changed my topic to. So some background on psychological safety. It was first introduced by Edgar Schein and Warren Bennis in their talk. Sorry, I don't have it written down here. Personal and organizational change through group methods. And in this study, they were looking at small group dynamics. So this is where the topic was kind of first introduced, but it didn't become very popular until Amy Edmondson in 1999 published her research titled Psychological Safety and Learning Behavior in Work Teams. And in this research, she looked at over 50 different teams in a manufacturing company and was trying to figure out how to measure psychological safety levels within those teams and their effectiveness. So she defines psychological safety as a belief that one will not be punished or humiliated for speaking up with ideas, questions, concerns, or mistakes, and that the team is safe for interpersonal risk-taking. So this is kind of a baseline definition of what psychological safety is. So from her research, the main takeaways that I got as like characteristics of psychological safety are that team members didn't have a fear that they would be blamed by others if they made a mistake. They could count on each other. Feedback was viewed as helpful, and they were reaching out for feedback. They cared about their work and believed that it was making a difference, and they had a shared learning behavior. A shared learning behavior meaning that they were learning but from what they were doing, from their errors and from their other experiences. So a real-world application of psychological safety in research is at Google, a company that we're all familiar with. So Google's known for studying their employees and trying to improve productivity. So they did a project, Project Aristotle in 2012, where they were trying to find the perfect team within Google and how to like improve productivity and have all of their teams be perfect or the most effective. So they looked at all different things: leadership styles, hobbies, their social lives of the groups inside and outside of the group, their education, background, personalities, whether they were introverted or extroverted. Sorry. And they didn't really find any correlation between any of these characteristics and what made groups more effective. So they kept doing research. They looked at a study that was done at Carnegie Mellon and MIT, where they took 699 different participants and threw them into random groups and gave them an assignment, some sort of project. And their most effective groups, the two things they found in common in this study were that everyone had their turn to speak, everyone was contributing in some way, and they all had intuition about how others felt. So they had high emotional intelligence and were able to understand how other group members were feeling. So both of these things you can attribute to psychological safety. So this gave Google their answer. They looked back with these two factors at their research, and this was consistent through all of their most successful groups as well. So this kind of became their answer on how to create that perfect group or the most effective group. So going into my research, I took what I had from Edmondson, and then from Google, what I took away was that they all understood the role of the team, that it was clearly defined, that they had their work had an impact and they understood that impact, that they were comfortable sharing divergent opinions, and they didn't have a fear of making a mistake or being blamed for their mistake. So these are like the biggest takeaways I got of how to like characterize psychological safety. So going into my research, the three things I focused on the most were whether they had a fear of making a mistake, whether they were comfortable sharing their divergent opinions, and how they viewed being vulnerable. And then I had kind of some more like supporter questions that included things like whether they benefited from their work and whether they could depend on their leadership. So this is kind of the biggest things that I focused on in my research. So going into my results, I had two different surveys. One was sent out to Westminster students and one was sent out to professionals in full-time positions. So starting with the one for Westminster students, I had about 11% of the student body was surveyed, and whenever they filled out the survey, they were asked to focus on the 1 to 2 student groups that they were the most involved in. So the main takeaways we got from this is that, so whenever you're looking at these graphs, the blue here shows a high level of agreement that they did not fear making mistakes, or a high level of agreement over here that they were comfortable sharing divergent opinions. So the blue is going to represent a higher level of psychological safety. So the class of 2023, being the juniors, had higher levels in both of these categories, and the class of 2025, being the freshmen, had the lowest levels, making sense because they haven't been here as long. I don't know, the seniors and sophomores fell in between. Does this have to do with senioritis? Maybe, I'm not sure. Without having more research, there's not really an exact way for me to pinpoint why the trends ended up this way, but this is what we found. Going to the full-time professional survey, 171 professional participants were surveyed. It was distributed over LinkedIn, Facebook, email. I had people from all different industries, including education, IT, healthcare, government, agriculture, accounting, and all over. And they were asked to focus on the work teams that they were the most active in whenever filling out the survey. So the trends that stuck out here were divided up by how many years the participant had been in their position. So for participants that have been in their position less than a year, again on here the blue is going to be higher levels of psychological safety. Those that had been in their position less than a year showed decently high levels. People from in the 1 to 5 year gap, it kind of dropped there. It increased from 6 to 10 years, and then the highest levels were those that had been in their positions for more than 10 years, kind of signifying that the longer someone was in their position, the higher levels of psychological safety and comfortability that they had in that group. So looking at all the participants and trends across all of the data, one question that was asked is if the participants had heard of psychological safety before. So for the professionals, 25% had heard the term before. For the students, 30% had. And the trend that we saw here is that those who had heard of psychological safety gave a more positive or neutral view of vulnerability. They were also asked to kind of define or explain how they thought of vulnerability. So those who had heard of it were more likely to give a positive view of vulnerability, and that showed in both surveys. And those who had not heard of it were less likely to give a positive or neutral view of vulnerability. So this kind of shows like a direct correlation between being aware of psychological safety and its benefits and being aware that vulnerability can be a good thing. So going on into vulnerability a little bit more to wrap things up, I did a lot of research like looking into Brené Brown as well, because she's widely known for studying interpersonal relationships and how groups and teams function together as well. And if you've heard of Brené Brown, you've probably heard the word vulnerability associated with her. So one question she does in her research that I read in one of her books is that she'll have people fill out the prompt, I grew up believing that vulnerability is. So this was also in my survey. And I put all the answers into a word cloud, and this is what we got. With the largest words being the ones that were mentioned the most often. So obviously the biggest one you see is weakness, and just underneath that you also see the word weak. Some of the other words that are up here, scary, hurt, afraid, bad, and there were several mentions of vulnerability not being masculine or not being okay for men to do. Other questions you'll see as well are growth, human, good, normal, and okay. So there were some positive views, but as you can see in this kind of infographic, the negative definitions and negative views of vulnerability are kind of what stuck out more widely. So wrapping up kind of my biggest takeaways from my research, and then things that you can take away from this presentation, hopefully to use in any of your work teams or student groups that you're involved in. The first one, to share your opinions, to know that your opinion is valuable, that everyone in the group's opinion is valuable. To admit your mistakes, own up to them, and to look for feedback so that you can build off of those mistakes and learn from them. That vulnerability is powerful. It's not a bad thing. It's a good thing to be vulnerable and to be able to kind of show your raw and true self in the groups that you're functioning in. And then to have an awareness of psychological safety. So hopefully if you came in here not knowing anything about psychological safety, even though this was a real quick and easy presentation, hopefully you've learned a little bit about psychological safety today, and you can go out and use the things that you've learned in your groups to be a better leader and a better team member. So thank you all.
Great, thanks to Anna for that presentation. And Anna will be happy to stay after if you have questions for her. She'll be right here. The last segment of this hour is to present our academic awards for the year. And so I have several people who will help us do that. The first is Dr. Heidi Levine, Director of the Westminster's Honors Program, and she will recognize the new members. Thank you. Thank you.
Great job, Anna. There are many ways to distinguish yourself at Westminster. It's my great honor and joy to work with the students who do that through the honors program. Many of those students will be presenting the results of their research today, and I'm really looking forward to seeing the fruits of your labor. Today we're going to recognize the students who joined the honors program this year for the first time as full year members. And if those students are in the audience and would please come down, I've got a fancy certificate for you. Gabby Scherer. There she is. John Stannard. And these students were also joined by Ayanda Huate and Fielder Aswai, who received their certificates last year. And next year, these students will be joined by 4 additional compadres: Joshua Morales, Tatum Covey, Trey Mazzarezzi, and Joseph Martin. Okay, congratulations to all of us.
We now have Dr. Don Holliday, Chair of the Biology Department, with the Cameron and Jessie Day Prize in Biology. Dr. Holliday. Thank you.
Good morning, everyone. Each year, my department selects one graduating senior to receive the Dr. Cameron and Jessie Day Prize. This fund was established by Dr. Cameron and Mrs. Jessie Day in 1961 and is given to the senior biology or biochemistry major with the highest GPA who has completed at least one course in physics and 2 years of chemistry. This year, that award goes to none other than Ashlyn LaFleur.
Now call to the front Dr. Ryan Bruner, Chair of the Psychology Department, for the Margaret McDonald Yeager Psychology Award.
Good morning. The Margaret McDonald Yeager Psychology Award is given to one senior psychology major each year to recognize outstanding academic achievement and the promise of successful graduate work. So the recipient will have compiled a minimum psychology course GPA of 3.5 and also demonstrated a strong interest in the discipline by involvement in learning experiences such as conference presentations, thesis work, and or internships. Since the purpose of the award is to assist a student with educational expenses, eligible candidates must have been accepted for graduate study. So this year's winner will be attending the University of Oklahoma to pursue a master's degree in special education with an emphasis on applied behavioral analysis. So the senior 2022 winner of Margaret McDonald Yeager Psychology Award is Rebecca Bond. And Rebecca's not able to be here with us today. She's actually in Hawaii right now, so she's probably experiencing a little nicer day than most of us. But the Yeager Endowment also allows 2 psychology juniors to be recognized for their early achievements in psychology. And these students receive funds to help with educational and research expenses. So the junior winners of the Margaret McDonald Yeager Psychology Award are Abby Nichols and Marley Richardson. Okay, I know there's some of them are joining us on Zoom, so I do have certificates for you when you are able to come get those. Thank you.
Thank you. All right. Now Dr. Heather McRae will present the Harman L. Rimmel III Prize for the Outstanding Thesis in History. Thank you.
The Harman L. Rimmel Prize for Outstanding Thesis in History is named after Harman L. Rimmel III, who graduated from Westminster with a degree in history in 1965. Rimmel was among the 7 Westminster students who gave their lives in Vietnam, and his family established this award in his honor. This year, the department is proud to award the Rimmel Prize to 2 students. First, to Brooke Thompson for her thesis, Killing the KKK, Lessons for Countering Right-Wing Extremism in America. Brooke can't be here today because she's honoring her commitment to the ROTC. The second student is Stephen Rogers for his thesis, How the Dreyfus Affair Changed Anti-Semitism in France. Congratulations to Brooke and Stephen.
Jessie Reed, coordinator of study abroad, will now come down and present the Piper scholarships. Thank you, Jessie.
Good morning, all. It was after very careful deliberation that the Study Abroad Scholarship Committee decided on 4 recipients for the Vernon W. and Marion K. Piper Endowed Scholarship for Study Abroad during the upcoming 2022-23 academic year. I don't know if you're here, you don't have to come down, but if you are here, please stand up. Dulcie Rosales Foster of Valdez, New Mexico, who will spend the fall 2022 semester studying environmental science in Freiburg, Germany through IES. Benjamin Parks, he's here, of Chesterfield, Missouri, who will spend fall 2022 studying finance and business administration in Norwich, England at the University of East Anglia. Allie Oglesby Watson, thinking of you are in here, are you? Of Macon, Georgia, who will spend spring 2022 studying Spanish language and culture in Oviedo, Spain at the University of Oviedo. And Kirsten Rushing, of Cedar Park, Texas, who will spend spring 2022 studying history in Aberystwyth, Wales at Aberystwyth University. And it's with great pleasure to know that Westminster College will be well represented abroad by some of our best students.
I have been asked to announce the Robert G. and Ruth H. Herber Scholarship for Classics, Philosophy, and Religious Studies. This award has recently been awarded to Lucille Vanek for the 2022-2023 academic year. Congratulations, Lucille. And now, Dr. Callie Wright-Smith, the Director of the First Year Experience, will announce the outstanding first-year students.
Good morning. Since 2020, we have awarded 2 nice certificates to our students to recognize the contributions of our first-year students. We award and recognize students for their outstanding academic performance and as outstanding first-year campus citizens, which basically means that they are contributing positively to their campus environment and providing a really wonderful Westminster experience for others. Nominations come from fall Westminster seminar instructors, and honestly, one of the best parts of my job is reading the wonderful words that our seminar instructors say about their students. They're very proud of them, they act like proud parents, and they talk really effusively about all the wonderful things that their students brought to their seminar class. Incidentally, this is the first time I've gotten to present this in person, ever, which is really exciting as well. So, for our first award for Outstanding First Year Academic Performance, we will recognize Samuel Rothermel and Kirsten Schrader. And finally, we will recognize as Outstanding First Year Campus Citizen, Michael Giles and Paige Brennicke. Thank you very much, and thank you to my seminar instructors as well for their wonderful nominations.
Okay, our final awards today are for the Undergraduate Scholars Awards, the Best Paper, Poster, and Presentation, which were submitted last week for the competition. And to give these awards, I would like to introduce the new director of the Center for Teaching and Learning and the new organizer of the Undergraduate Scholars Forum, our own dear Dr. Mary Majerus.
Thank you, Carolyn. It's my distinct pleasure to announce the awards for the best papers and posters this year. This was a very competitive year in determining the winners, so congratulations to all who entered. You've done remarkable work. In the best paper category, the award goes to Lucille Vanek for Self-Reliance Through Gandhi and Deep Ecology.
Go Blue Jays.
And congratulations for the best recorded poster. The award goes to Emily Nelson, Marley Richardson, and Maya Westfall for their poster Stress, COVID, and Alcohol Use. So I'm just going to take a quick minute here. This is a special year, and the Committee for Teaching and Learning would like to thank all the students and their faculty for their contributions to this. You are what makes this event exceptional. I do want to especially highlight the participating seniors this year who have led academically in bringing the USF back in person. As a token of remembrance, we want to share a small gift with you, and I want to explain this briefly. This glass sun catcher has the historical Westminster logo inscribed on it, noting Westminster's commitment to knowledge. It was crafted by the Blanco Glass Company, which, not coincidentally, is the company who has created the unique windows for our Westminster Churchill Museum Church, St. Mary Aldermanbury. This token is given in hopes you will continue to seek knowledge, remembering your time fondly at Westminster, where you connected and grew with students and faculty in our learning community. So seniors, please come down at the conclusion of this session and pick up your sun catcher. However, before we conclude, I do have a few more brief notes. As we conclude, I want to thank the key players in the USF who have worked so diligently behind the scenes. Special thanks to our presenters and their dedicated faculty sponsors, the incredible IT team, the marketing communications teams, the teaching and learning committee, student ambassadors, the Watt committee, and the Education Association. Deepest thanks goes to the Stubbs family for their generous support of the Scholars Forum each year. We are so grateful for the support and for attending us in this very special year. And I'd like to pause to give special thanks to an individual who is responsible for a great deal of the success for the Scholars Forum since its inception, Dr. Carolyn Perry. Dr. Perry was instrumental in creating the vision of what this forum is today. She led the way early on, envisioning USF as an incredible opportunity for students to learn from one another. She shepherded us through the COVID years, smoothly transitioning this outstanding event to an online forum. Each year, her leadership has kept this forum a strong and vibrant academic event. Please join me in celebrating the long-standing legacy Dr. Perry leaves this institution through her continuous support of the USF. Thank you, Carolyn. I think she should say a few words, don't you?
Well, thank you. I used to always start this day saying, this is my favorite day of the year. And students used to think that was pretty funny, but it's the truth. And so thank you all. I've enjoyed this so much over the last 15 years. And so I would just like to thank you students for being here to support each other and for, again, the great presentations you will make today. Thank you for being a great audience this morning. Be sure to sign in to your sessions so that we have a record of your attendance. And now, please go grab a donut and some coffee and enjoy the rest of your day. Thank you.